Well, let's get started on this last one. So the title of this is Chernobyl, Environmental Effects and the Dichotomy in Radioecology. What's a dichotomy? A division. So there's a divide in radioecology. Not all radioecologists are agreeing on a certain aspect of this. In fact, there's a lot of debate. There's a lot of division in the science of radioecology right now. And this talk will tell you a little bit about that. And it's related to Chernobyl, which you have all studied, and many of you have evidently been there. The radioactive releases went on for 10 days, contaminated 200,000 square kilometers, and thousands of people were evacuated. One of the main themes of Chernobyl is controversy. Regardless of what topic you choose, you can generally find opposing views if you look in the literature long enough about the value of evacuating the public, for example, or the quantity of the initial release, or the cost, or you just name it, ecological effects, human mortality. Some things in the literature say there were thousands of people who have died. Other things in the literature say 24 firemen died. So controversy is the key, or a key, to the Chernobyl accident. In 2004, the IAEA established what's called the Chernobyl Forum, and they recognized all this controversy, and they said, let's get all the experts together and come to a mutual agreement about a lot of these controversial topics so that the public is not so confused. So their goal in this Chernobyl Forum was to reach an international consensus and eliminate the controversy about the effects of the Chernobyl accident. They published a report. That report did exactly the opposite. It caused a lot more controversy. And I'll mention that to you in a minute. The Chernobyl Forum was a very impressive array of organizations and participants. This is a list of all the organizations which had representatives in the Chernobyl Forum. The World Health Organization, International Atomic Energy Agency, United Nations Development Program, Food and Agricultural Organization, the United Nations Environmental Program, Office of Humanitarian Affairs, the United Nations Scientific Committee on the Effects of Atomic Radiation. They had representatives from each of the three most impacted countries, Belarus, Russian Federation, and Ukraine, as well as 80 other experts from 12 countries. So it was a powerhouse of knowledge that went into writing these reports and reaching a consensus. All these people had to agree before the findings went into the Chernobyl report. So I'm going to give you the results of that from an environmental perspective. But first, I'm going to tell you a little bit about our knowledge about radiation effects before Chernobyl. I'll then give you an overview based on this Chernobyl Forum report. And that overview will take different perspectives. One is the temporal aspect, that is, the effects as a function of time after the accident. I'll also talk about general effects to major classes of organisms. What were the general effects to plants? What were the general effects to animals? I'll also talk about indirect effects and confounding variables. And all of this leads to this current controversy that I've discussed that has caused a divide in radioecology. And I'll tell you about that divide and give you some examples. So this is before Chernobyl. This is that graph now that you've seen 3 times. So we knew a lot about the acute dose and the lethal dose for lots of different plants and animals, and that mammals were the most sensitive. We knew that because they had been studying it for 40 years, 40 years before the accident. You know, I started this with that health physics publication, all that good solid data. So before 1986, when this accident happened, there was a lot of data that had been produced. The data came from laboratory experiments, accidents. There was an accident in 1956 called Kashtim, from areas of natural high background, from weapons fallout, and from large-scale field irradiators. This is an example of a field irradiator. Inside here is a very high-level cobalt or cesium source. You can control the source by raising it up or lowering it down into the ground. It irradiates the environment in this direction. You can measure the dose as a function of distance from the source, and you can look at the effects as a function of dose. This is the Pawnee grassland in Colorado. They had similar irradiators in all the major ecosystem types. So they had one in the forest site, grasslands, tropical, mountain zone. This was a major funded program to look at effects on the environment. And a lot of good data came from this before Chernobyl. Oops. So I showed you about acute effects, but there are also, before Chernobyl, a lot was known about short-term exposures from 5 to 60 days. And this chart breaks it up into minor effects like chromosome damage or change in reproduction, and intermediate effects, so mortality of individuals within a population, but the population still remains viable. And here we have different categories again: the coniferous forest, rodents, soil invertebrates, etc., tropical rainforest, and we've got minor effects and intermediate effects. And here on this scale is dose on a log scale. So these over here are the most sensitive to radiation, and these over here are the most resistant to radiation exposure. Now, I show you this because I'm going to change this scale and use this chart several times in the talk, and I'm going to overlay the Chernobyl effects data. So you'll be able to compare Chernobyl to what we knew before the accident. However, the Chernobyl data is not generally reported in total dose, but dose rate, grays per day. So all these studies were 5 to 60 days. I'm going to convert this chart by arbitrarily saying that rather than 5 to 60, they were all done at 10 days. I'm going to divide this graph by 10 and make it grays per day. I haven't changed anything other than dividing this scale by 10 and converting it from a dose to dose per time, grays per day. Now we've got dose rate. Now I can overlay the Chernobyl data, and you'll compare it to what was known before. So part of that Chernobyl forum was a group that looked at the environmental effects. Alex Sakin is one of the best radioecologists in Russia, and there's a whole suite of other people involved there. Okay, so these are results from the Chernobyl forum, and one of the most important statements, and a statement that is not considered adequately when you talk about Chernobyl, is the effect of time. And that when you look at the environmental effects, there were 3 periods of time. One was the first month right after the accident, then from month 1 up to about the first year, and then the third zone is year 2 to the present. This is the relative contribution of dose. And you can see that the dose rate drops off, or the percent of dose drops off very, very quickly. So this says that the majority of the dose was acquired in the first month after the accident. It decreased rapidly, and now, from year 1 to current, the percentage of the total dose is less than 1%. Also, the biota were exposed to a diverse group of radioisotopes. There's tremendous heterogeneity, and the accident occurred at peak sensitivity of many biota. The accident happened in, April 26th. That was a time in early spring, and organisms were very sensitive. Okay, let's look at this first component, the first month, the first 20 to 30 days. There were severe effects to the biota those first days. Gamma dose rates were 20 grays per day. We just discussed dose rates in Fukushima currently being less than 40 micrograys per day. So 1,000 times less. During the first 20 to 30 days, dose was dominated by many short-lived radionuclides. This is a graph of time after the accident, day 1, day 10, 100. This is the contribution to absorbed dose, 60%, 100%. And you can see that these radionuclides, the contributions are mainly from these short-lived radionuclides. This is the purple one here is the graph for cesium-137 and 134. You can see that at 10 days, its contribution to dose was only about 10%, and it took more than 100 days, probably 5, this is a log scale, so probably a year and a half before the contribution of cesium was significantly greater than the contribution of these other radionuclides. So this first 20-30 days where dose is so high is largely due to these short-lived radionuclides. Radionuclides that were not emitted largely from the Fukushima accident. High dose to thyroids from iodine was a major contributor. Here are the air exposure rates. This is the reactor. This is the town of Pripyat. This is a scale of 1 kilometer, so that's about 3 kilometers. Look how quickly the dose rate decreases. Here it's 20 grays per day. Here in the town of Pripyat it's 0.02 grays per day. This is still, this is short, this is on the 26th of April. This is still a tremendously high dose, but the point is the heterogeneity and how quickly it decreases as a function of distance from the reactor. So, this is the graph that I made for you a few minutes ago. Let's plot these two things on here and see how our pre-Chernobyl data would have predicted effects. So if we put those down there, we can see at these dose rates you would expect minor effects to rodents, minor to intermediate effects to these sensitive organisms, whereas the moss and lichen, the radio-resistant ones, most likely there were very little effects. This first phase, there was acute effects within the 10-kilometer zone. This is the red forest that you've heard about before they cut it down and buried it. Radiation levels were sufficient to cause mortality to the trees. So there was a community level effect. Reproduction effects early on, lots and lots of mortality, lots and lots of negative effects. Okay, the second phase is from the first month to the end of that first year. This period is characteristic of a drastically decreasing dose as a function of time. And the reason is the decay of the short-lived radioisotopes. This is time after the accident. These are months, 3, 6, 9, 12. You can see the contributions and the decay of these short-lived isotopes. Cerium-144 was basically gone after 10 months. Ruthenium isotopes gone after 16 months. So their decay caused this radically decreasing contribution to dose. Likewise, decreasing effects. Also, there was a lot of migration of radionuclides into the soil, so animals were not being exposed as much during this period, and the contributions of beta to gammas were changing. This third phase is from year 2 out to the current. Dose rates are now chronic, and they're less than 1% of the initial dose rates. So if you go there now and you see effects, or if someone says there were tremendous effects from Chernobyl, you can say yes, but what time period are you discussing? Shortly after the accident or now? The probability of effects now are very different than the probability of effects shortly after the accident. This time sequence is important when you discuss effects of radiation at Chernobyl. Also Fukushima, because of the 134 cesium declining. Beta to gamma contributions are now comparable. It depends on the bioaccumulation of cesium. Cesium and strontium now dominate, whereas all those other short-lived radionuclides dominated early on. Indirect effects dominate, genetic effects persist. So I just told you a temporal component that the Chernobyl Forum identified and published in their proceedings. Now let me tell you a little bit about the general effects to plants and animals. And we'll compare it to that graph of pre-Chernobyl data. Here's morphological mutations occurred at 1 to 15 grays. This is a pine tree, and this is called leaf gigantism, where these follicles grow much, much larger than they're supposed to. This is an effect of radiation exposure in pine trees. Hardwoods were more radio-resistant. This is a pine tree that shows damage up here. Right here is an aspen tree, a hardwood. It doesn't show any damage at all. So there was individual species variation. There was a shift in ecosystem structure. The dying pine stands were replaced by grasses, and then a slow invasion of these hardwoods. Genetic effects were documented, and they extended in time. Here's work by Vasil Yochenko. He's at the Institute of Environmental Radioactivity. He did a lot of work at Chernobyl, and now he's doing work at Fukushima. He says one of the typical morphological changes in Scots pine is due to this thing called canceling apical dominance. This is normal growth of a pine tree. It goes like this, with this central trunk just continuing to expand each year. If these trees are exposed to large amounts of radiation, you cancel this apical dominance and you get this type of growth. So that central trunk branches and it no longer exists. This is typical damage to pine trees. Here are examples in the forest at Chernobyl. You can see it there and there. Here is a very old tree without the central branch and it divided. Okay, at 0.3 grays, growth and developmental problems. Again, we would have predicted that. Inhibition of photosynthesis occurred, transpiration, chromosome aberrations, short-term sterility, and high mutation rates. Again, 0.3 grays per day is a very high dose rate compared to what Fukushima is experiencing today. There was lots of research on this plant called the Arabidopsis. Its genome was characterized very early. They measured mutation rates in these plants as a function of time. Now I'm going to switch to general effects to rodents. In the fall of '86, rodent populations in the exclusion zone decreased 2 to 10-fold at dose rates of 1 to 30 grays. Here's those dose rates. Here's rodents pre-Chernobyl, and yes, they would have predicted such severe mortality based on pre-Chernobyl data. At 0.1 gray, there was infertility and reduced testes mass and several other effects from the radiation. In the third period, 1 year to current, the data on the rodents are contradictory. You can find data in the literature that says that there are still effects on the rodents, and you can find data in the literature that says there are no effects currently. Part of that ambiguity is due to the fact that now many generations of rodents have evolved. The dose rates are much lower. It's chronic exposure. Some of the studies were inadequate from a dosimetry standpoint. Many different effects, many different parameters could cause this disparity in results. Soil invertebrates received a tremendous dose because of the radiation falling on the plants, being washed off, and then infiltrating into the soil. So the soil organisms received a very, very large dose. And populations of soil invertebrates were reduced 30-fold, and reproduction was strongly impacted at these high dose rates. At 30 grays, it altered the community structure and species diversity in soil organisms, and that altered community structure changed for 2.5 years before it recovered. This is something that would not have been predicted. This is 30 grays. Here we are, and here are the soil invertebrates. We would have predicted moderate effects, whereas a change in community structure is way out here. So our pre-Chernobyl data would have mispredicted and under-forecast the effect on soil invertebrates. Genetic effects have been documented. There are some suggestions of genomic instability. Some epigenetic work is now being done and documented, and suggests that chronic low-level radiation might alter the genetic structure of populations, increasing the karyotypic variability in the offspring. All right, now we're going to talk a little bit again about indirect effects of human abandonment. You've seen this slide before, and you've seen this one where wildlife flourished at Chernobyl, according to some. This is, again, data from the Chernobyl Forum report. All this is just from the report. They said that after the accident, many years after the accident, they actually introduced 2 species into the exclusion zone. The European bison and this endangered Przewalski's horse. Both populations are doing very well in the Chernobyl zone now. These animals were at one time documented to have existed in the Chernobyl zone. They disappeared. After the accident, they reappeared when humans left. Lynx, owl, swans, possibly a bear. I've seen lynx in Chernobyl myself, so I know that animal exists. These animals became much more abundant, as well as these species of birds. So, let me summarize a little bit what the Chernobyl Forum said, and then I'll discuss this dichotomy, this division in radioecology that you should be aware of, because it affects how you view the literature that you read. So in summary, when you look at the Chernobyl accident, according to the Forum, they mention this 3 different periods. The first month, there was tremendous radiation levels, tremendous mortality, in most organisms and populations that you looked at within the 30-kilometer zone. The second period, according to the forum, is from 1 to 12 months. Lowered dose rates, its morphological effects persisted, and the soil invertebrates were severely impacted as the radiation went down into the soil. So much that community-level effects were observed. The third period is after 1 year to the current. This is a period of ongoing recovery from radiation. There are secondary effects, noticeable positive effects from human abandonment, and there are long-term genetic consequences that are still being studied and currently unknown. So, main conclusions from the forum. Radiation caused acute effects. These are quotes right out of the Chernobyl Forum report. Increased mortality, reduced loss in plants and animals. It took a few years to recover from major radiation, induced adverse effects in populations of plants and animals. And then, the Chernobyl Forum wrote the following thing in their final report. They said this: due to removal of human activities, the exclusion zone has paradoxically become a unique sanctuary for biodiversity. This is a direct quote from all of those influential organizations that composed the United Nations Chernobyl Forum. Immediately, this hit the press. Right after the report came out, this is in the BBC News, wildlife defies Chernobyl radiation. It contains some of the most contaminated land in the world, yet it has become a haven for wildlife, a nature reserve in all but name. However, the next year, scientists said the idea that the exclusion zone around the Chernobyl nuclear power plant created a wildlife haven is not scientifically justified. And from that, a division started, a dichotomy in the radioecology community, with one group saying, yeah, there's wildlife there, they're doing okay, and another group saying, that's wrong, you don't have scientific evidence of that. We have the scientific evidence, said this group. Our data shows drastic impacts. These people said, but wait, the doses that you're reporting suggest that the effects, you can't be seeing those effects at that dose rate. So there was a lot of controversy and debate. For example, here, work by Mueller and Mousseau. This was reported in 2009. Reduced abundance of insects and spiders linked to radiation at Chernobyl 20 years after the accident. 2 decades after the explosion, Chernobyl nuclear power plant, radiation is still causing a reduction in the number of insects and spiders. Now, because you guys are now radiation ecology experts due to my single lecture, you should know that what? Insects and spiders are radio resistant or radio sensitive. Remember these charts that I've been showing you from mammals up. Who says insects are more sensitive than humans to radiation? Who says insects are more resistant to radiation than humans? Yay, resistant. What about the time period? 20 years. The Chernobyl Forum stressed the importance of this first month, this first year, and the time after. This is 20 years later, and these scientists are saying that there are still impacts to some of the most resistant, radio-resistant organisms on our planet. So the radiation ecologist, the traditional one, says, wait a minute, this is not what we know. This is a new paradigm. If this is right, we need to change our thinking in radioecology. All that pre-Chernobyl data doesn't make any sense. This is brand new. This is hot. Is this right or wrong? Let's look at their data. This is those insects. This is micrograys per hour. These are the insects that they studied. Here's their data. I can't read that. Let's see. Oh, these are the numbers. So that's the number of grasshoppers, the number of bees, etc. This scale is log, 0.1 microgray per hour, 1 microgray per hour, 10 micrograys per hour. In Fukushima, we're 6 micrograys per hour. We just heard a moment ago. Now, let's go back to that chart that I showed you, and let's plot these data on that scale. These data are in micrograys per hour. Our data has to be in grays per day. So here's 0.1, right here, micrograys per hour. We do this little conversion, and we come up with this 0.1, 2, 3, 4, 5 zeros grays per day. Where does that fit on our scale here? Wait, that's 0.001. We've got to add all these other zeros in. Let's see. Let me move my chart. We'll do that, that, that. So, pre-Chernobyl, here's our soil invertebrates. These are minor effects. These scientists are saying major effects 20 years later, not here, but way down the scale here. Here's the dose rate of their study. This, by the way, is the IAEA guideline for acceptable levels of radiation in the environment. So the classic radioecologists who studied data prior to Chernobyl questioned this manuscript and said, how can you document radiation effects at such low doses? These are background doses. If there was such major impacts at background levels, wouldn't we have a decline in insects the world over, not just inside Chernobyl? So this report on insects 20 years, the decline in insects 20 years after the accident, deepened that division in radioecology. And that division continues. And it's driven by these two scientists, Moeller and Mousseau. And they are very, very prolific. 84 Chernobyl-Fukushima manuscripts since 2007 in very, very good journals. Better journals than I publish in. Nature, Science, Evolution. They are writing the new book on Chernobyl effects. But all of their data are controversial and counter to what we knew before Chernobyl and counter to other studies that other scientists are doing now at Chernobyl and Fukushima. So we have a divide. We have some people publishing that there's major effects, other people looking at these and saying, wait a second, there's some problems here. They just published a work on cataracts in rodents, and it was again in a prestigious journal. Here's the link. I can email this. Well, you have it. I encourage you to link to that and then read the review afterwards. And the review is very harsh criticism of the technique that they use. They captured rodents and then took the animals and put them in a freezer, and then 6 months later examined cataracts, clouding of the eye in these animals, and they got a tremendous number of percent cataracts in the eyes of these rodents, the mice. I think it was 70%. The people who criticized this study said, if you put eyeballs in the refrigerator, it causes them to cloud, and you don't have any controls. So the methodology of some of these studies is questioned. There's an interesting link there that I encourage you to call up. It's a story written by the BBC. It describes this dichotomy. It talks about Mueller and Musso, and it talks about the other scientists who are questioning their work. So not only is there a divide in radio ecology, that divide is so apparent that the British Broadcasting Company is writing stories about it. Potential causes of this controversy are poor dosimetry, the spatial heterogeneity of the exposure, confounding variables, lack of appropriate controls, questionable statistics, what constitutes a significant effect, and perhaps even motives beyond science. The point of all this is, do not believe everything you read. You are learning to be a scientist. You are learning about radiation. When you read something, just because it's published in a journal doesn't make it true. Just because it's published in Nature or Science doesn't make it true. Question everything you read. Question everything that I've said today. Don't believe me just because I'm up here talking to you. Be critical. Be a good scientist. Formulate your own conclusions based on your scientific training. Ask people that you respect and you know, and you'll be a better scientist. Whew! I'm done. Okay, questions of that. Wait, we have to have a test first. We have to have a test after each one of these. Let's see, true or false? I read this article in Nature, and because it's in Nature, which is probably one of the best scientific journals in all the world, I know it has to be fact. I can believe it. True or false? True. Just because I say it's false doesn't mean that you have to believe it's false. Maybe you still believe that everything you read is okay. You know, when I learned that, well, early on, I was told, don't believe everything you read. At that time, I was reading newspapers and such, and I would take them to someone, what's going on here? They said, don't believe everything you read. In the back of my mind, as a new scientist, I thought, phew, thank goodness, I can still believe in science. I can read a science article and I can believe it because of this process, the scientific process. I do work, I publish it, or I submit it for publication. Other scientists review it, they criticize it, I have to change it. If it passes criticism, it's published. And that's another step. After it's published, people can comment on it afterwards. That's a strong way to do science. It's our scientific method. I'm so glad that that exists because now I don't have to believe the newspaper, but I can read a science article and by goodness I know it's true because it's published. Then I started reading things that I questioned, and maybe this isn't true. This is counter to what I've learned. Now, I'm very sad to realize that even it's in science, you need to question it. And there's a lot of stuff, a lot of junk that's published nowadays that passes the review process. You've been good.